Hi, I'm Matt, lead developer on IDPicker. In this tutorial, I'm going to introduce the protein view. If you're familiar with IDPicker 2's spectra per protein by group table, this is the equivalent. In the default layout, the protein view is in the pane at the top left. It provides a view into your data that is protein-centric. Each row in the table is a different protein or protein group. You can see this little alpha helix icon, so clearly it's a protein. Before explaining what the columns mean, let me point out the summary counts in the pane's title line. It shows how many clusters, protein groups, and proteins there are in the current view, as well as the protein level FDR. The key column, the one that must be unique, is always on the left and is always visible. For proteins or protein groups of one protein, it's simply the accession of that protein. For multi-protein groups, it's all of the accessions of the proteins in that group combined together. That can get very long, but it's what we do instead of picking an arbitrary representative or anchor protein. The next column is the cluster ID, which is assigned according to the same kind of arbitrary clustering that was in IDPicker 2. It exists to show which protein groups share peptides with other protein groups. The next column is a brief visual representation of sequence coverage. You can hover over the cell to get the numeric value and resize the column to get more detail. There are 4 different colors representing the depth of coverage for residues at that location. The lightest color has no coverage. The second lightest has 1 distinct peptide. The next has 2 peptides. And the darkest color has 3 or more distinct peptides. The next column is the protein group ID, an arbitrary integer identifying the protein group that it belongs to. It's a way to refer to the protein group without that long list of protein accessions. The next 3 columns are counts associated with that protein. Separate counts are given for distinct peptides, matches, and spectra. Finally, there's a description column, which is everything in the FASTA that is not part of the accession. All of these columns, except the accession, can be rearranged, hidden, and customized from the Display Options dialog. From here, I can set the number of decimal places, the foreground and background color for each column, and the visibility. Auto means that the column may be automatically shown or hidden depending on whether they would be redundant or irrelevant. You may be wondering where the individual columns per source group are, a la IDPicker 2. Those extra columns can be enabled from the Pivot Options menu. You can pivot either by source group, source, or both, and you can do it for distinct peptides, matches, or spectra. Now I can see individual columns for each group and source. The source columns have actually been trimmed to only show the unique part of each source name, if it's possible to do so. The basic unit of the protein view represents, unsurprisingly, an individual protein. If you want to group proteins by their protein group and/or cluster, you can very easily configure the tree grouping. You can sort each column by clicking on the header cell, and change sort order by clicking it again. The Find box allows you to search for text within the protein view. The text can be part of the description or the key column. You can even paste in a list of accessions to filter for. Remember to press Enter when you're ready for the filter to take effect. If you quote the find string, the entire cell contents of one cell must match the quoted string. Like all IDPicker views, the protein view can export its data seamlessly to the clipboard, a tab-delimited file, or directly to Excel. The first 3 export options will export all visible cells. Columns hidden by display options are not exported, by the way. The last 3 options will only export selected cells. That's it! Now you know the power of the protein view, and knowing is half the battle.